All right, so let's now move on. I would like to discuss, today we are moving into more and more into biology. I would like to discuss now telomere. So I will explain what are telomeres. I would like to discuss a model about telomere change across cell division. And again, it falls into this framework of Markov, not Markov process, we're going to have Markov jumps. So let me state the question and the biology. So telomere dynamics across cell division. So this we started a couple of years ago, the following question. So you know that when a cell is born, it can divide a certain number of times. So this is cell, let's say time 0. I call time 1, the different time I increase the time when the cell divides. So it is known that after a certain number of divisions, the cell cannot divide anymore. This is called senescence. And that's it. The cell do not divide. As you know, in cancer cell, this threshold, this event can sometimes it do not happen correctly, and the cell continue dividing. And this is why cancer cells start to invade and create all the damages. So the process by which a cell stops dividing is not fully understood. It's under real investigation, especially what are the mechanisms that are involved to tell a cell not to divide. For example, for yeast, you can have a certain number of division, some 60 division, and after some time it stops. But there are variability. Sometimes you can see a cell that can have 90 divisions and everything is fine. So what defines the threshold by which you stop dividing? So let me remind you that 2012, the Nobel Prize in Medicine was given to for answering this, partially this question. And the key answer to answer this question was the length of the ends of the chromosomes, which are called the telomeres. So you have basically in the nucleus, you have the chromosomes that are the organization of which is still unclear, but basically they are encoded here. This is called the centromere. And here the ends here are called the telomeres. So I would like in the class, the next time or the time in 2 weeks from now, discuss that actually in yeast, these telomeres, you have 32 of them. And when people ask, where do you see them? Where are they? Well, the answer was they are located on the surface of the nucleus, and more important, they form clusters. Now, these become a very interesting questions in clustering that allowed us to revisit all the Swolochovsky equation for clustering, because what people found is that they could form a cluster of, you could see 4 to 5 clusters in each cells. And the question is, why do you have clusters? What is the mathematics of clustering?
So David, that's known. The telomeres are always fixed to the surface? Or?
No. This was it was for yeast, and it was for, let's say, some kind of normal conditions.
Right.
Now, people did experiments during quiescence, that is when the cell is under stress. What happened, and so, of course, you can, if they mutate a certain protein, which is called the SIR protein, it's called SIR3, if I'm correct, then this are not anymore attached to the surface. They come inside, and inside what people have found, and this is still under investigation, they can form a big cluster, a huge cluster. And again, those properties like coagulation, fragmentation, they come, they bind, and they can unbind. Of course, what you have to understand here is what is the meaning of forming a cluster? If you have two telomeres that comes, they can one is coming, a second one is coming. Now, how do you put the third one? Is the third one going to bind to the two of them? Or would you have something like this, that they come as the finger in your hands, they come nearby like this. So when you have a break, you just break uniformly like here. Or if you have four, you can bind, you can dissociate this way or that way. Or so this you have to make assumptions. But I won't discuss this today. I wanted to discuss this. Then you can study here a model of coagulation fragmentation with 32 to ask the following question. Suppose you have Brownian particle on the surface of a sphere. You have 32 of them. They are moving by Brownian. If they come, they can collide. You wait. What do you see? What's the steady state? Is the steady state a single ensemble of telomeres? Or do you have severals? And if you have severals, what is the mean size of the cluster or the mean number of telomeres per cluster? And there was also an important question, is that if telomeres can exchange genetic information, then you would like to ask the following question. What is the time two telomeres are going to spend in the same cluster? It's like the pair correlation function. So if you have, let's call this is number 1, and this is number 3. How long are they going to stay together? While many events can happen, because you can have a telomere that was nearby, that went, and went off. So the cluster may change, the dynamic of the cluster, its identity is changing, but you don't care, because what you care is really about having the two specific that you have in mind in a cluster. So this was a tricky question in combinatorics. So I'll postpone that for later. All right, I'm explaining where the problem is coming from. So you have these telomeres, and now I'm not discussing the spatial component. I'm now going to just forget about the geometry and discuss about the temporal aspect of it. So what happened during a cell division? During a cell division, if you have the end of the telomere, the end of the chromosome, so this is the chromosome, and this part is called the telomere, so it's more fancy, it has some kind of shape. I'm not going to enter into the details of the structure. And the idea is that if you are at stage K, when you go at stage K plus 1, with a certain probability, this has been diminished. The number of base pair here has been shrinked. So this is what can happen usually. But there is a process in nature which does the opposite. So this is more or less, this, you can see this event as systematic. Each time you have a division, you shrink. But there is a rare event which involves a protein called the telomerase. And what this does is that it can increase the size significantly. So this is like the usual, but sometimes, rare events, you have a big one. And of course, this is, you can think now, if you have this, this will maintain the length, this prevents the length to shrink. Why are we talking about the length? Because, and this is why this Nobel Prize was given, it says that basically what people were looking for years is that the clock of a cell is maintained by the length of these structures, which means that you shrink, you shrink, you shrink. If you reach a threshold, as we discussed before, if you reach a threshold, that's it. You stop dividing. There is a process which is unclear, unknown. I mean, probably it has to do with all this molecular pathway inside the cells. That the cell knows that it has reached the threshold and it will not anymore continue dividing. There is no next generation. All right, so this was our starting point. So let's write a model of this to study, for example, there's one more thing that I have to add here. Which telomere are we talking about? Is there one specific telomere? So for example, in yeast, you have 32. So they all have their own life. That is, let's assume that during this process of division, you have the 32 of them, and what happens to them are independent from each other. So you can see them as independent processes, and either a telomere can have its length decreased, or sometimes, rare event, it is increased by this telomerase enzyme. So then came, and it's still not fully resolved, and this was the interaction with our biology partner, is that the cell should be sensitive to the shortest one. That is, you look at your 32 and you are interested in the shortest. Because if the shortest has basically started to eat its genetic information, that's it. The cell doesn't want to reproduce if the genetic information has been damaged. You don't want to do that. That's why during double-strand DNA break, and the cell has broken, the DNA is broken, if the DNA is not repaired, the cell stops whatever it has to do. It just has to repair. And if it doesn't repair during a certain time, the cell triggers apoptosis. It's better for the entire body that this cell dies than it replicates with abnormalities. This is the biology. Biology is good to kill, to preserve the ensemble. All right, so now we started to look at this question by saying, all right, let's model this, and this is what I would like to present now. I'll introduce the model, and then during the second part, we'll make a break after this, we will look at the analysis. So what is the model? The model is to look at the lengths. So let's forget now that you have 32, because we said that they are independent. Let's look at what happened at the length of the telomere at division n+1. So think about a Markov process. You look at the division, the length just after division ln. And what can happen is that it can decrease. I have to have the right... it can decrease with a fixed length a after each division, with a probability 1 minus p that depends on ln, or it can increase, and now this is a random variable. It can increase, and the mean of this variable is much bigger than a. So the probability of this psi equals y, we took it as an exponential law, py. And so I'll give you the values. So the values here are given for yeast. For other organisms, they are probably different. So because we worked on yeast, we had to find out all of these values for yeast. So p, the p here, which is your exponential parameter, was equal to 0.026. And so the mean number of base pair that was added was about 40 base pair that is added. While A here, in average, it was 3.5, between 3 and 4. A was of the order of 3 to 4, so we approximated it at 3.5. With probability 1/2, it was those guys. So this is about 10 times. So this is really a much bigger growth than the decay. And now, what was interesting is that we could find from data this PL. So we had points. So the probability of one of these events to happen was just decaying, that is the probability, the PL, when you shrink, this probability is increasing. So when you reach this state, basically the cell wants to prevent you to go drastically to shrinking without getting a chance to elongate. So the further you come to death, basically, the more chance you have to survive. And actually there are diseases where the telomere is affected, and it leads to accelerated aging in people. So this is a very hot topic to understand biological and genetics. What are the genetics disease associated to telomeres and cancer? This is very hot. Of course, we're not going to cure cancer by integrating by parts, but the idea is to give some understanding about how you analyze the data, what's the model, what can you predict, what happens when some parameters are changing, because sometimes it can affect with drugs some parameters in the model. All right, so for P of L, we just fitted, this is fit, you take data, you just fit a curve, and we fitted 1 plus 1 plus beta, L minus L0 for L bigger than L0, just, elementary function, and 1 otherwise. And beta here was, again, I give you parameter because usually nobody cares about parameters, but I think it's in the parameters, especially when you see small and large, that you can do then the calculation, the approximations. And L0 was 90 base per. All right, so I think with this, I'll give you one more information about, with already this, you can calculate, for example, what is the mean length. That is, suppose you wait and you have steady state, you can calculate the mean L minus 1 minus Ln. You can average this. So now you know the probability for this to happen. We know the distribution for this. And basically it's minus L multiplied by 1 minus PL plus 1 divided by PL. And so from this, at steady state, when you're not growing and not diminishing, so Ln plus 1 equals to Ln, you can get an average length of 350 plus minus, I forgot what, I cannot read anymore what I wrote, I think 102. All right, so now what are we going to be interested in? Suppose, all right, we have the model. We have information from data. Basically, all of the parameters now are fixed. There is no unknown, no model to fit. We got everything from wherever we wanted. Now, let's wait. You start, let's suppose you start with an L0 and you wait. What's going to be the steady state distribution of the lengths? Where are they going to be, how are they going to be distributed?
Can I ask you a question?
Yeah.
That L equal 350, is that a stable point that if you start there, it sort of stays there? And is it a stable point? Is it
stable? So, that's right.
So it's stable to small perturbations.
That's right.
Okay.
That's right. But usually you start, let's say, you start with a very long telomere, and then
I mean local stability.
Yeah. So when you arrive here, this is stable. When you're here, you don't move. It's stochastic. So here at this point, you go around and then you come back. Basically, when you reach in this region here, you have some kind of. This is where you find the steady state. You have a balance between decreasing and increasing. Although you have to imagine your process, you decrease all the time, and then you have a big jump. So there is this large fluctuations, but this is the mean field steady state.
And there's only one
yes.
fixed point?
Yes. Okay, so now here are the questions that I'm going to write down the questions, and then we will do the analysis after the break. So here are the questions. So what is question number 1? What is the steady state distribution? Second question, what can we say about the distribution of the shortest? So what is the mean length? Mean length of the shortest. So shortest among the 32, right? Among the 32. And now, so the debate in the field was the following. Is it true that this is the shortest? Why not an ensemble of the first, of the second? So we thought about doing the following calculation. Not that it answers exactly this question, but we said, is it possible that there is a gap, a statistical gap? You know, it's like when you solve partial differential equation and you look at the eigenvalue of your operator. You can have what is called a gap between the first eigenvalue and the second one. And usually this gap is interesting because it captures some inherent properties of the system. So we said that if you have the shortest, so this is the length here. And so you look at the first, the shortest one, which is here. Is it conceivable that if you look at the mean and if you look also at the variance, you have a region here, an area, and this area is it's separated. That is, what does it mean separated? It means if you calculate the distance between the shortest is the mean length of the shortest, and the mean length of the second one. That is, this is number 1, and now this is number 2. That you have indeed a separation, that this guy doesn't fall into the fluctuation of that one, because if it does in the fluctuation of that one, then it could be more complicated, that the cell might not be waiting that the first one has reached a threshold, but probably to the first, second, third, an ensemble of them. So what I'm going to show with the statistical calculation at the second half is that indeed there is not a spectral, but a statistical separation between the mean of the first minus the mean of the second. This clearly separated, and the variance is smaller. This is what I'm going to show. So there is some kind of separation, right? So we will discuss this, and there is a third question that I don't have time to answer. I will not answer in this class. Is what is the mean time to a threshold? Because now it becomes a mean first passage time problem where you impose a threshold at some point in this jump. So you have a jump process and you want to calculate, to estimate, when are you going to go over this, let's say you fix a number, what you think is critical. How long do you have to wait? And how this time depends, for example, on B. So the parameter beta here accounts for the effect of the telomerase. So if you change beta, it's like changing the concentration here. So really all parameter can be connected to the biology. All right, so let's break here. And after the talk, I'll show you how you can solve this in the continuum. This is very well-known technique. It's called the Takak's equation. This is a very well-known approach that has been used in queuing theory for the last 30, 40 years. And I'll show you that you can have an explicit expression for the distribution. And we will calculate using Laplace methods the mean length of the shortest and the second one, and we will see the separation. All right, so let's break 10, 15 minutes. We'll have to come back, and I have to leave at 4:45 because I have to pick up my kids from school. All right. Polymer, which is bit connected by spring, this is alpha equals 1/2. Usually for diffusion, we have 1, here we have 1/2. And so here's the question. If you prescribe alpha, this is prescribed, this is 0.43120. Is there a polymer model, and how do you have to build a polymer model where you prescribe this exponent? This is given. Why this question is becoming important, because we had to analyze all the tracks of single particle trajectory, and we could find there was a spectrum of exponent. Now the question is why? So to look at this question, we decided to reinvestigate Rouse polymer, theory of polymer, and see how do you have to connect a single monomer with long, not necessarily the neighbors, but with longer ones, so the idea is to have an algorithm, given alpha, tell us what are the connections. So that with this connection, so we're talking about long-range connections, not anymore neighbors, then you create this anomalous exponent.
So this is a sort of distribution on the length of the segments.
That's right. So now you have, this is, instead of having, let's say, a factor, let's say a certain number for this one, you modify the one here, and now you start to connect with the second neighbors and the third neighbors, and they're fixed. They are fixed. You fix them forever, and then when you look at the exponents of this guy, not the extreme because they behave a bit different because they don't have connections, but if you look at what happened inside, you can see that the alpha is the one you prescribe, and I will explain how you construct this.
And presumably you could have many constructs like that? Or?
Maybe. We know how to offer one.
Okay.
If you have others, probably there are many ways to do that.
So it's some optimization procedure?
I will show. I will go step by step.
Okay.
It's about calculating variance of the motion of this and see how it depends on the neighbors, and actually the procedure is you diagonalize the matrix and you use the eigenvalues, and instead of having the eigenvalues of the Orstein-Uhlenbeck operator with a certain power, we change the power and see how what's the consequence of the connection. And this power is exactly, some function of this power is exactly what you get in this exponent. So we could then go back and change the power here and explain how this is reflected in this long-range connection. I would like to discuss this. I'll try to go to this today. And again, this class, the next class, and the class after, I would like to spend really 3 classes about the nucleus organization, telomeres, clustering, chromatin organization, how do we analyze single particle trajectories, conditioned on the fact that you know that where you're observing lives on the polymer. It's not just a particle that is Brownian particle, it's something that belongs to a chain. Now, if you know this a priori information, you should be able to use this information for your prediction about what kind of alpha you're expecting, what are the forces you are expecting. Another question that I would like to discuss is, suppose there is a force on this one. There's a force. You observe this one. You observe the trajectory of that one. Can you, from the trajectory of that one, reconstruct the force that was exerted here? So obviously you have to deconvolve between what happened between the monomers in between. So this is the second question that I would like to go into all of these type of questions. It's 2 to 3 more classes.